Meeting is being recorded.
All right, welcome everyone to the ESIP Soil Ontology and Informatics Cluster Call. It is October 18th, 2022, and today we're going to hear a presentation from Erin Berryman from the US Forest Service about how the forest inventory assessment. I'm sure that I'm butchering the acronym. Oh wait, so forest inventory and analysis deals with geolocations. So thank you very much for coming to talk with us, Erin, and take it away.
Great, thank you, Kathy. Can everyone still hear me? Thumbs up?
Yes.
Okay, we're good to go. Fingers crossed that the technology gods smile upon us. I'm Erin Berryman. I'm so happy to be talking to you guys today. Just a little bit about me. I do have a soils background. I have a master's degree in soil science, but then I got my PhD in forest resources and really consider myself more of a forest ecologist. So I have been working for the Forest Service in some capacity for about 5 years now, and most recently, the past year as part of the Forest Inventory and Analysis program. So we do have a little bit of a soils program, as you'll find out, and as you may already know, but a lot of what I'm going to talk about at first applies to the entire FIA program. And then if we have time, I can go into some specifics about the soil and litter collections that we do. But focusing on the geolocations. So really briefly, the FIA program, we are responsible for the National Forest Inventory here in the United States. And we have all lands covered in CONUS, at least. And Hawaii is a recent addition to our inventory. Alaska, we do not have completely spatially covered, but we're getting there. So Alaska is still running off of the periodic inventory where we don't get to all lands in a single rotation, but it's the nature of Alaska. It's a little bit different, a little bit big and challenging. But everywhere else, lands everywhere and any ownership and every part of the states, you will find an FIA plot somewhere. So we are tasked with keeping that inventory current. This is just some mandated language here in this first slide. It's not just about national forest owned land, right? So we make it for everybody. We create this data for everybody, policymakers and federal, state, tribal, and local government, land managers, landowners, investors throughout the forest sector, and also non-governmental groups. They actually are some of the biggest users of our data, are NGOs. So just a brief overview of the structure of FIA, because it will become important later, because there are a few regionalized differences in how FIA does some things, but overall it is a national program. So I am in the Rocky Mountain FIA, and it's 8 states that we are responsible for. There's 4 regions, and they are unified under a common framework, a common sampling grid, but we may differ in some of the add-ons that we do. And soils is actually one of them. Every region collects or has collected soils in some respect, but they do differ in the intensity and how often they do it. Here in the Rocky Mountain region, we do collect soil at every single one of our FIA plots. So we do have one of the more comprehensive soils program. Although it is kind of in its infancy. So moving forward, we expect to have be building up our soil samples and soil data pretty quickly. Okay. So before we talk about the nuances behind releasing the geolocations of FIA plots, a little bit of background about how we decide where to locate those FIA plots. So the FIA grid, it's hexagonal. You might recognize it here on the left. It's the large hexagons. We're defined the Forest Health Monitoring Program, which FIA has existed in one form or another, going all the way back to the 1930s, but it wasn't national, it wasn't consistent, it wasn't anything close to what we have today. The Forest Health Monitoring Program, which started in 1988, was really the first glimpse of what the FIA program has become, because the Forest Health Monitoring Program was really the first nationally standardized monitoring program. We didn't really call it forest inventory at that time because it just wasn't, it was more tasked with monitoring forest health than it was actual inventory. But that really did lay the groundwork for the modern FIA program. And part of that groundwork is this FHM grid. So those bold, so this is actually a snapshot of the actual grid. So the bold lines, the bold hexagons, the bigger ones, that is the FHM grid. So in this, I think about approximately in the center of each one of these hexagon cells, there was an FHM plot, about one plot every 160,000 acres. Now, when FIA was told to integrate with the FHM program and really become a truly national annualized program, we started with this FHM grid, divided each larger hexagon into 27 smaller hexagons, and then we gave one hex, one plot. And that plot is randomly located within that hex. So it's not necessarily in the center of each hexagon. Today, about 350,000 plot locations, CONUS wide. Now this is just plot locations. It doesn't mean that every single one of these locations is forested, but there is a location there and we do track something on it, even if it's a barren desert, even if, or I shouldn't say barren, even if it's barren land, even if it's road, water, whatever, we have information about what's on that plot location. So it's a lot of data. Now we get to why we cannot tell you exactly where the FIA plots are. Okay, so the law tells us that we have to maintain security around these FIA plot locations. If you want to know the details, you can go to this Appropriations Act from the year 2000. Basically what it does is it pulls from an earlier act that applied to a lot of other things, but not specifically forest inventory, but now in the year 2000, they made it official. Forest inventory plots, we have to protect confidentiality of those plot locations with the intent to prevent the link of individual landowners to plot information. So some of the stuff that I'm going to talk about only applies to privately owned land that FIA plots are on. However, because of the confidentiality requirement and the framework that we use, what ends up happening is all of our plots get applied some level of fuzzing. And so none of the latitude and longitude locations that you can find in the publicly available FIA data set are actually true locations of any plot. There are strict, it's maintained pretty strictly. And in fact, right now, FIA has a moratorium on releasing any plot locations because we do have certain agreements, like especially if we're in the same agency, if we're inside the Forest Service, we can create agreements to actually release the plot locations. However, there's a moratorium on that right now because we did have concerns about some of those locations getting out. So we do try to maintain that confidentiality pretty closely. But in addition to that, even if you're not just talking about this legal requirement, we do have a responsibility as a program to provide unbiased data, and it can be compromised if people know where these plot locations are, right? Users can intentionally alter the plot. They can, even if they have the best intentions, you know, maybe they're trying to protect a plot for forest monitoring. We don't want to have any involvement. We need to be able to monitor this as unbiased as possibly. And we also have a lot of privately owned plots, or I should say plots that are on privately owned land. And that trust gets built up over time and it can get eroded if there's any doubt that the private landowners, that people might know where plots are located on their land. So, so there's a lot at stake here that we need to protect. Oops, wrong one. Okay, so how do we protect plot locations? The Forest Survey Handbook lays this out. We use a process called fuzzing, although some people prefer to use the word perturbed. I don't think it's as fun to say as fuzzing, so I say fuzzing. Fuzzing and swapping plot locations are all fuzzed. They are fuzzed by randomly changing the plot coordinates by up to a half-mile radius from the actual location in any direction. And it turns out we do have a few plots that are fuzzed up to 1 mile. It's a minority of plots by far, but that does have to happen in special cases where maybe half a mile isn't quite great enough. A small number of plot locations must be swapped with other similar plots in the same county, and these are privately owned plots. Masking must be used on all FIA raw data from private lands that is publicly released, whether it's from plot owners, primary wood using mills. We actually have another part of our program that doesn't get talked about a whole lot with ecology, but it is mill survey. And of course, all of that's privately owned. And so there's confidentiality requirements with that too. Now, swapping is not required for data from public ownership, but fuzzing must be applied. Okay, so a little bit more detail on that. So plots on public land, we do have this documented in, there's a couple of references that I'll put on the last slide on this. So fuzzing only. Most plots are fuzzed within a half a mile. Like I said before, a very small percentage are actually fuzzed within 1 mile, and there's no way to know what exactly you have with any given plot. Now, the fuzzing is constrained within county of origin. So nothing's going to land outside the county that you're interested in. And this is especially important so that it doesn't impact our standard FIA estimation techniques, which we tend to like to summarize things by county. So we can be rest assured that this plot is going to be somewhere within the actual county. Oops. Now, for the plots on private land, we have the fuzzing like the public land plots do, but we also introduce swapping. And what the swapping does is creates uncertainty in plot ownership. Roughly 3% are swapped with what we call like plots. So not all of them are swapped, but a small percentage are swapped within the county of origin. And what that means is that the plot ID is going to be swapped so that you cannot link, in the public database, you cannot link that ID with that actual plot. Now we have a separate database. We actually have 2 other databases where we retain behind the Forest Service firewall the precise, accurate linkages. So we can always do that crosswalk, but as far as the public facing data set goes, those plot IDs are swapped. So what does a like plot mean? I think this is a really, really important question, depending on your application. It does vary by region. So remember there's 4 FIA regions that I showed at the beginning here. Most of them do require that the same forest type is used and the stand size in one way or another, whether that be stand density index or density, some sort of measure, or basal area, some sort of measure of above ground similarity. Okay, now I think the big question, and unfortunately is still largely unresolved, especially for questions of soils, is how's the integrity of these data impacted by fuzzing and swapping? To be honest with you, there's really little research that's been done on this, and it's really unfortunate given how many research projects are actually using fuzzed spatial locations. Even studies that link up the fuzz spatial locations to remote sensing data, for example. Now, many of them have done it successfully, so there doesn't seem to be a huge impact. There was a paper, McRoberts et al., 2005, that came out 17 years ago for study areas, and that's because this is when this fuzzing and swapping procedure first came out. So we've been using this for 20 years. It hasn't changed. So for study areas greater than about 20 miles radius, there were very negligible impacts on FIA estimation of attributes using the FIA design. And so what that means is, for example, our county level estimates of tree density or stocking or carbon available, those values were within the margin of error, whether you swapped it or didn't, or fuzzed it and swapped it or didn't. So seems to have negligible impacts there. Now, where you start to see more impacts is where you're actually intersecting that fuzz spatial location with spatial data sets to build a model, for example, a predictive model. And that's where a lot of the remote sensing studies, you'll notice, will really try to work with FIA to use their precise locations to get those data, especially once you're working with the spatial scale of Landsat data, for example. You really are going to see an impact of using a fuzz location over a precise location. But like I said, this hasn't really been studied very much, and we don't have great guidelines for how big of a spatial scale do you need to go before it's not an issue, for example. So I wish we had better answers to that question. And let's see, I think that might be, oops, that is. So that's what I have for the geolocation stuff. And I do have a few slides on the actual data collection involving soil and litter at the FIA plot locations, but I thought I would pause here and ask for any questions regarding the geolocations. No questions, okay.
Go
I
for
don't
it.
see anything in the chat.
Can I go for a question?
Oh, yes.
Hi, it's a great introduction to FI. Thank you for the presentation. I was just wondering, this is something that I know of how much, I mean, I'm a soil science person, so I was wondering how different is it from a typical NRCS soil survey data? I think something in addition to soils data, do you have something that has some information on the tree species or something else in this forest inventory and analysis?
Yes, so, and that might be a good segue to the data collection here. So the plot layout you can see here is the 4 subplots that are relatively close to each other, about 50 feet apart from each other. And it's the same in these circle footprints, the trees are all inventoried. So in the white circles, so in the small circles. So every tree is measured, standing or dead species. We record things about condition of the forest, disturbances, canopy cover, crown condition, understory, seedlings. We don't have very detailed non-tree data, but we have some general cover data. So we know approximately what the understory composition might be like. We also have a forest type. A forest type gets assigned as well. So we have, and some aging, some tree ages. So we have stand age. We have pretty much every forest attribute that you can think of, we have recorded on these plots. And directly below those trees, we have collected the soil and the forest floor. So we can spatially directly associate the forest attributes to the soil samples. And I'm not nearly as familiar with the NRCS procedures as I am with these procedures. So I can't really say how they're different. Besides that, I'm pretty sure they don't do a tree inventory. So but there might, in terms of vegetation surveys, there might be some additional information that NRCS does. I'm not sure, but does that answer your question?
Yes, it does. Thank you.
Okay, great. Yeah. Oh, and one thing I didn't I didn't get into is how often the plots are visited. Because we have so many plots, we cannot visit them very frequently. They get measured once every 10 years in the West, for the most part, although some states are starting to visit them more frequently. And out East, they are measured roughly every 5 years, 5 to 7 years.
Are the plots across years linked to the same identifier? So can you follow a single plot with that geolocation fuzz throughout the time course?
Yes, you can do that. Yep.
And Tanya, I thought I saw you unmic and maybe have a question.
Yes, actually what you're showing right now, Erin, actually answers quite a bit of it. I guess the other thing would be, so sometimes in water resources, they will purposely like study a site for 3 years and then they'll pick up and move to a different site and things like that. I guess my question is, do you ever do longer term? And then the other question is, because you said you tend to visit these sites every 5 to 7 years, say on the East Coast, do you visit all of them within a region at the same time, or do you try to make sure that regionally you're getting a snapshot in a given year?
Yes, the latter, yes. That is the idea. So if I wish I had a diagram of that, but if you can go back to this hexagon here, this grid pattern. So we would sequentially number them. If you can imagine the center of each of those little hexagons, 1 through 10, and then start over again. And we would go in and measure all of the ones 1 year, and the next year would be all of the twos. So the idea is that you would get that larger 200,000 acre-ish area sampled every year, just in a different location. Does that make sense?
Yes, it does. I mean, that's great.
Okay.
That means
I should have put a
great
diagram
information.
together for that. That's a really good question and very important question from estimation standpoint as well.
Thank you.
And then I think, oh yes, so you talked about long-term. Yeah, so that's the idea, is that these are permanent plots. Once they're there, they are visited forever in perpetuity. Occasionally, very, very, very occasionally, they do get moved out of just extreme necessity, right? But that's always very well documented, and it should get assigned a new plot number. So we would have that old plot record start to fall out, and it won't be measured again, and then there will be a new plot that will pop up. So that's something that we try to very carefully account for. But yes, the idea is that these are permanent plots and they will be revisited. We do have, you can look in the database, and actually that's one project that we are working on in RMRS. A fellow analyst of mine is trying to link up some of the historic state inventories, because a lot of the historic state inventory programs were sort of converted to this FIA program. And in many cases, because it was a different plot design, it didn't really work out for them to do a direct conversion, but in some cases it did work out to do a direct conversion. And so we're working on a crosswalk to where we can find plots that have been visited since the 60s in New Mexico, for example, which is going to be a gold mine of data. And I hope that we can release that soon, that crosswalk.
Last question, I guess.
Yeah, go ahead. Yep.
So is there any talk of putting active monitoring at any of these sites?
I wish. I would love to, but do you mean like sensors?
Yeah, like I'm a
Mhm.
little obsessed with soil moisture or
Oh, yeah.
something like
Yeah.
that.
I know. Wouldn't that be fantastic?
Yeah.
I would love to. Large scale. It's resources, it's cost and resources. It's going to be a hard sell to do it everywhere, but we are always not necessarily able to fund everything, because it does vary year to year depending on what our budgets look like and what travel got canceled, which there's been a lot of it lately. But we won't go there. So there's always room for pilot studies, which is really, really cool. One thing that's really great about FIA is that it is regionalized. And so the different regions actually have quite a bit of autonomy when deciding how to run their program. And when deciding, they might want to do an add-on, like maybe the Rocky Mountain states decide that it is well worth our resources and time to monitor soil moisture changes throughout the year, for example. Then we could probably scare up some funds to do a small pilot study on a few plots and do that. So just putting the word out there, I'm always interested in stuff like that. So give me a ring if you have interest in trying to set something like that up. All right, cool, great questions. Yeah, so besides all the tree stuff, we have a soil sampling program, and we call, FIA is funny, we call forest floor a soil sample. The forest floor is sampled at, so first of all, every plot looks like this, this set of 4 subplots. That's the national design. The white area is where the trees are going to be measured. And the yellow area is just this broader zone that allows us to sequentially sample forest floor, pull big samples away, presumably without causing a ton of disturbance because it's nice and spread out. So that's the idea with those transects that you see here with the little hashes. That's for, well, it's for the soil as well, but it's really more for the litter, so that's far away from each other. So we collect forest floor every time we visit plots in the Rocky Mountain region states, which is the 8 states in the West, except for the ones that border the ocean. So no California, Oregon, or Washington. There's a 30 centimeter diameter sample. That's all pulled away. We total carbon and nitrogen bulk density on the litter, and water content. And water content is just obviously the water content at the time that it was collected. That's basically it for the forest floor. Now we do archive all of these samples, so we still have them sitting around, and occasionally somebody will pull some of those samples to do an add-on analysis. So we do keep everything. Now the mineral soil, we only go down to 20 centimeters. We do 0 to 10 and 10 to 20. And we use a really nice impact sampler. It's 5 centimeters diameter, and it retains the volume pretty well so that we can get a bulk density value from that. But the spatial replicates are terrible. We only have one that's collected at every plot, and it's only collected at that subplot too. So that if somebody had to, if I were forced to say what the one worst limitation of our soils program is, that is the first thing that I would say, is that we only have one replicate, one spatial replicate per plot. Field texture is assessed, or texture is assessed in the field, and then the sample is brought back and analyzed for total carbon and nitrogen, bulk density, water content at the time of sampling, coarse fragment content, so at least we got that pushed in, so that was good. pH, extractable phosphorus and sulfur, and exchangeable cations. And we also probe the soil depth up to 50 centimeters. And that's what we have for the soils. It's they're analyzed very consistently. We have, I think each region has a lab that they send their soils to. So there's only, I think, 2 or 3 labs that are even really processing all of this. So that's all pretty consistent. And the biggest challenge right now with getting that data is the backlog of analysis that we're starting to see. So I think that we got in RMRS, we got our 2013 soils just loaded to the database like earlier this year. So it's a pretty big backlog, but it's expected to pick up. We've just had to hire some people to do it.
Can I
Now,
ask a question?
yeah, go
about
ahead.
So when you
Yep.
say forest soil, forest floor, is that your litter layer? Is that the organic layer?
the
Like,
whole, yeah, so they pull everything down to where they can distinguish a mineral soil line.
Okay.
Things get a little less straightforward in organic soils. We don't have a ton of those. Well, at least not out here. That's more of an issue for out east. But they do. It's, yeah. I think the training for collecting wetland soils is a little less straightforward. They are told to. I think the manual says, the field guide says something like, in the case of highly organic soils, your mineral soil sample becomes your organic soil sample or something like that. And it's like, well, that's not really that clear, but.
Okay, so
Yeah.
this 30 centimeter diameter tries to capture the organic, the above the mineral organic horizon, and then the 5 centimeter sample tries to capture below that mineral organic horizon.
Correct. Yes. Okay.
Cool. Thank you.
Yep. Yeah.
So one other question about sampling. So it's my understanding then that wherever these 3 circles that you're showing here are in space, essentially those are permanent. I think that they're trees, but maybe I'm wrong. Or they're oriented around trees. Maybe I've misunderstood that. I guess my question is, how is it? Yes, I'm not sure.
So yes, there's trees. Are you talking about the white circles, the smaller circles?
Yes.
That's where the trees are measured, correct.
Okay,
And the trees that would fall out in that yellow area wouldn't be measured.
Okay.
Yeah, so they're just measured on those 4 subplots.
Okay, so you're measuring your soils away from the trees that you're measuring.
Correct. Yeah, outside of the subplot.
Okay,
but close.
But close, right?
Yes.
So my next question, having worked in sites that are relatively small and permanent, how do you keep track of where you've already sampled?
Yes, so that's what that little train track looking thing is supposed to help with. So the first visit, you're going to sample right in the middle. The second year that you visit, you're supposed to sample right next to it. So you follow the 1, 2, 3, 4, 5, 6, 7, 8, 9. So theoretically,
okay, I'm sorry. Yeah.
No, that's okay. I did not explain that very well.
Okay,
Yeah.
I was like, did I miss that? Okay.
Yeah. So the idea is, we have 90 years of soil visits that we could make before having to figure something else out. And
can you remind me what the diameter of these samples are, or of these plots are, of these circles that you're presenting?
Yes. So let's see. So there's larger samples. So there's a 10-foot. It looks like it's 10 feet in between the samples. So now the subplots themselves, I believe, I always forget because we have different P&Ws as bigger ones. I think we have 8-meter radius, 30 feet radius. Yes. And so you can see how in the soil visit number, train tracky looking thing, so that would be a total of 90 feet across, or 80 feet. 1, 2, 3, 4, 8, 80 feet. There we go.
Cool. Thank you.
Yeah.
It seems wonderfully well organized. It's quite impressive, given that they're permanent. I mean, I don't mean to say that I would have expected otherwise, but clearly you've anticipated a lot of these issues and you have a plan, and that's pretty great.
I think really that's good to hear. There's definitely been some criticism of this program when they first started thinking about soils. I think it's been tough. Anytime you get the forest inventory, forest biometry world is very small and is steeped in tradition. And when you try to bring that world together with soil scientists, interesting things happened. So I heard, but I was not in the room for these conversations. So it's not bad. I will say that we have lots and lots and lots of data. And I think the value really comes in our numbers. in our sheer numbers because we have so many locations where soil has been sampled exactly the same way. I think that provides a good advantage for this program. All right, any more questions about the soil protocol here? I did have a couple more slides, but I think I've really already presented these, and I don't know how many of you saw these, but this is about the down woody material, which is soil adjacent, but it is something that we also measure on those same plots. So I don't know if anybody was here for that presentation. That was back in, was it July?
Please repeat yourself.
Great. Okay. So we do have down woody material sampling. And now we do this using a transect method. So this comes from the world of fuel monitoring, Brown's transects, where you just record the intersection of the pieces with the measuring tape in certain diameter classes. So it's broken up into 4 components: the coarse woody debris, which is the stuff that's bigger than or equal to 3 inches in diameter. And because we are foresters, we go with inches. Fine woody debris, we have 3 diameter classes, 1 to 3 inches, 1/4 of an inch to an inch, and then less than 1/4 of an inch. There's also litter depth. Now, litter depth is, so forest floor depth is collected when the litter sample is collected with the soils. Okay, so like back here. So we know what the depth is, and it's I think that's collected in 4 different locations in that forest floor sample. But this is going to be a litter depth at more places in the plot, so that we get kind of an overall view of what the litter depth is at this particular plot. And this is also where we distinguish litter from duff. Whereas duff would be included with the forest floor sample, here we are measuring litter separately than duff. And this is just a depth measurement. Nothing is taken away. The Transect design has changed a few times over the past few years, and so that will have a big impact on the calculations and how you use that data. But currently, this is what that design looks like. From the center of these subplots, we have transects radiating out, and we have 2, we call them 2 transects radiating out from the center subplot. It's kind of weird because when I first saw this, I said, oh, there's only 1 transect per plot. And I was corrected and told, no, there's actually 2 transects per subplot radiating out from the center. Okay, yeah, so that's what we have. And that's visited every time we go out as well. That's also an RMRS thing. So I don't think other regions measure the down woody material every time they visit a plot either. And how to access that data, FIADB, if you need help accessing any of the publicly available data, we do try our best to make it publicly available. And I know that we fail sometimes. So email us, we'll point you in the right direction. But you can download our data a variety of different ways. We have SQLite databases, which I love working with. They play friendly in R, but we also have CSV tables, and we might have an access database as well for somebody to use if they really wanted to do it that way. But yeah, so that's what I have.
Thank you. Let's open the floor for some brief clarifying questions, and then we'll roll into an open discussion. Does anyone have any clarifying questions? This is where I sit on my hands and try not to say things. All right.
No question is a stupid question, and I glossed over a ton of detail here. So if you're not familiar with FIA and you want to know more, just feel free to ask right now or later, whatever.
Okay. Oh, what is DWM?
That's a great question. That is down woody material.
Nice.
See, this is the type of stuff I don't even notice as it's coming out of my mouth, right? I'm so used to acronym speak.
All right. On that spirit, thank you very much for your presentation. We will post
Oh, of
this to the
course.
YouTube channel, and I'm going to hit stop the recording now.